Okay, so am I audible? Um, can just check like and reply to the poll. Okay, so is the audio clear? Yeah, okay. So first of all, welcome to Drug Discovery. We have with us Dr. Jagdish Mahale, who is a pharmacology expert. So he will be guiding us today for our session. So before we start, I'd just like to let you know that this will be an interactive session. So whenever at any point of time, if you have a doubt, you can just raise your hand, and after that, I'll take you on the stage, and you can just ask the question, and you can again go back to the audience. Okay. So I request Jagdish sir to take over.
Okay, thank you, Devin. Can we have the presentation on the screen, please? Nikhil, can we have the presentation on the screen? Okay. Hello, everyone. Good evening, all those who have joined. So, am I audible to everyone? Can you just give a like or something? Excellent. Thank you. So today I will be talking on drug discovery process. Uh, I come from a pharmaceutical background, so that explains why I'm talking on this topic. So I'll just start by introducing myself. Next slide, please. So I have a pharmaceutical sciences background. I did my Bachelor of Pharmacy from University of Pune. And then I did my Master of Pharmacy with a specialization in pharmacology from North Maharashtra University. After that, when I was still looking for my PhD, I did like two very short jobs, one in Nasik as a clinical trial coordinator in Shatabdi Hospital, and one as a drug safety officer in Mumbai for Roche Pharma, but I was actually working for TCS. And then I had already got a PhD offer during that time, and I started my PhD in 2012 at University of Leicester. And my specialization in PhD was cancer studies and investigating natural cancer drugs for their remedial properties against lung cancer, which is what I was researching on. And then after I finished my PhD in 2016, I immediately was offered a postdoctoral position at University of Oxford. And since 2016, I have been at Oxford. Now I'm working on a different aspect of cancer research. I'm more into cancer immunology, which we can discuss after the end of the topic. So can I have the next slide, please? So I'll just like to give you a brief overview of my talk. So I'll just go and start by talking from like the very beginning, like how did the evolution of medicine started? Like what was the medicinal practice in old age and how it has evolved over the centuries. Then I'll go into a drug discovery process. And in drug discovery process, there are various phases like preclinical studies, clinical studies, regulatory approval. So it's actually a very long process, but I will be today focusing mostly on preclinical studies and clinical studies because it can be a very exhaustive topic discussing all these subjects such as product launch or regulatory approval itself is a very big and vast subject. So I'll just today focus on the evolution of medicine, preclinical studies, and clinical studies. Next slide, please. So The medicine has been around for a very long time, and maybe it started like 5,000 years ago, where which we consider Ayurveda as the most ancient type of medicine. Along the same timeline, there were other system of medicine as well, like Egyptian medicine, Chinese medicine, or Unani medicine. So this evolution and discovery of medicine, it goes back very long time, like 5,000 years ago. We'll just start looking at what were the practices of Stone Age and how did the discovery of medicine actually started. Next slide, please. So Stone Age, as you can see, there was no actually medicine available during the Stone Age. People around that time, they lived generally only 25 to 40 years. There are a lot of common causes that people died during those times, where this is from the archaeological excavations that the scientists have found out, like arthritis, infections, wounds, tooth disease. This is how people mostly died during those times. And what were the causes? At that time, the people believed that there is evil spirits or angry ancestor. This is what causing the disease. There was nothing like, they did not know that it's something because of a natural cause. It's something because of a pathogen or germs or bacteria that actually cause disease. So they just thought like it's evil spirits or angry ancestors. And what were the actual causes? Actual causes were they did not have enough diet or varied diet. The nutrients that they should have received for their body to function, they never got them. So diet and not having varied diet was one of the reason, and also the cleanliness and the hygiene part came into effect, and that's why they actually had a very short lifespan. And treatment, there was obviously no treatment available. Like people used to think that okay, we will do some religious ceremonies, some animal or human sacrifices, charms, spells, amulets. That's what the people at that time believed in the treatment, which was obviously not true. So then we will progress into the stage of Ayurveda. Next slide, please. So, among all the ancient medicine, obviously the most well-known ancient medicine systems are Ayurveda, Chinese, and Egyptian medicine. But for me, I think even at that time, the most developed system of medicine was Ayurveda. Uh, Ayurveda was the first system of medicine actually to propose that diseases are due to natural causes. There were no such medicines, but the saints at that time, they discovered the natural power the nature gives from like trees and herbs. Like they had this curing ability. So this is one of the thing that the first drugs that came into effect, when they were all natural origins, they were all herbal. And they actually developed a lot of theories, like what are the reasons that actually people fell ill, like the three doshas, vata, pitta, and kapha, which are mostly metabolism movement related disease. So they actually had a proper diagnosis and a prognosis around 5,000 years ago. And also what they discovered is that medicine is like, there has to be specialty like we have in today's time. There is a cardiologist, there is an oncologist, there is a doctor specific for arthritis, there is an immunologist, there is even a pediatric doctor just specialization for kids. So that is one of the things that they had developed, like each and every type of disease, they have to be treated by different doctors. That was one of the principle. As I said, they had developed a very um, uh, system of diagnosis, like even which are still used today, like checking the pulse, urine examination, checking the tongue, checking the stool examination. This type of examination, even the doctors, even the modern age of practice, they are still followed. And in terms of treatment, it was, apart from herbal medicine, they developed a lot of other treatment, such as digestion, and doing yoga, exercise, meditation, use of herbs. So those ancient saints, like even though like this is 5,000 years ago, this kind of treatment, like such as doing exercise, yoga, getting sunlight, we get vitamin D from sunlight. So this kind of things or theories which we developed 5,000 years ago, and without doing much scientific experiments, they still hold the truth. And the first book of medicine which was ever written was by a sage called Sushruta, and it was one of the most exhaustive uh, a collection of all the types of diseases available and all the treatments that are being available. So can we go to the next slide, please? Apart from Ayurveda system of medicine, there were other system of medicine like Chinese medicine and Egyptian medicine. But the thing is, with the Chinese medicines, they had what differentiated the Chinese medicine is they had herbal medicine, but they also had animal medicines from animal origin. And they developed a lot of other ways of treatment, such as massage therapy, acupuncture, cupping therapy, skin scraping. But obviously, these things could not be um proved scientifically, such as massage or acupuncture can actually help curing your disease. The Yes, you may feel better, but as per scientific experiments, these things have not been proved yet. And one really developed another system of medicine was at that time was Egyptian medicine. The Egyptian system of medicines, they were the first in the world to have proper doctors and surgeons and pharmacists at that time, like 5,000 years ago. They had studied the human anatomy very well. They also had developed a lot of herbal medicines. They developed the practice of having good hygiene, good health. Also, uh, sorry, I'm just saying, like, is everyone able to hear? Is everyone able to hear properly? Okay, I'll just carry on. And if you have any problem hearing, just like let the moderators know, because I'm just seeing some comments that they are not able to hear very clearly. Okay, so the Egyptian system of medicine, one thing that a lot of people used to eat raw meat during that time, and they also developed the theory of you should have cooked meals, which helps improves your digestion, and all these again were very exhaustively documented during that time. So this is what how like the evolution of medicine actually started 5,000 years ago. Yeah, next slide. Next, yeah. What we call now this as the age of modern medicine. Now this is like 400 BC, like around 2,500 years ago. And what we call as the father of modern medicine today is Hippocrates. And Hippocrates was a stalwart of those times. One thing that was he was hugely successful in is that he separated religion from disease. Uh, he could convince people that the diseases are not caused due to evil spirits or gods. There are natural causes such as infections, germs, bacteria that cause disease. And at that time it was very difficult for people to convince those things. So that was his one of his biggest achievement. And then he went on to describe over 200 different types of diseases such as cancer, mumps, measles, diphtheria. Like this is an outstanding achievement during that time, that he was able to describe so many diseases like 500 years ago. He also described this mental disorders, that it's an actual disease and it's not an evil spirit or it's not a punishment by God, but the mental disorders are an actual disease, and they can be treated with help and support and medication. He was one of the first people to start a school of medicine. He made medicine as a professionalism, that the doctors need to be honest. They themselves have to follow a code of conduct. So he made practicing medicine as a professional. It gave he gave it a professional outlook. And even the doctors today, when all over the world, when they finish their medical degrees, they take something called an Hippocratic oath, which are some of the basic principle all the doctors should follow into their medical profession. So he was truly a great figure. So can we have the next slide, please? Okay, then how did the evolution of medicine started? Okay, from uh, Hippocrates, he was very um, successful in describing a lot of a lot of type of diseases. But how did further evolution happen? One of the major things that happened was Leonardo da Vinci. He was the first person to dissect a corpse and he drew very detailed drawings of our human anatomy and human structure. Where are all the organs located? How many bones are located? How they actually function together? So Leonardo da Vinci, he made a very detailed drawings of our human body, of our human anatomy. And then there were a lot of other inventions that actually pushed the discovery of medicine, and which included invention of microscope, then other organs and how they function was eventually discovered in 17th century, like motion of heart, a description of blood vessels. And then in 18th century, Edward Jenner, a British scientist, he was the first person to discover first ever vaccine against smallpox. uh, that happened in 18th century. In 19th century, there was nitrous oxide, which was discovered as an anesthetic. Louis Pasteur identified germs as a cause of disease, and so on. And then one of the most important discoveries ever in the history of medicine was the discovery of structure by Watson and Crick in late 1940s, 1950s, and that really changed how the medicines were discovered and how the drug development happened. So, like getting the structure of DNA was one of the most significant discovery in medical science. Can we have the next slide, please? So, drug discovery process. How do we define drug? What is a drug? Drug can be any substance, natural or artificial, which is used in composition or preparation of medicine. And there are various phases in drug discovery process, as you can see here: pre-discovery, lead and target identification, preclinical studies, clinical studies. I'll go through some of them today. Can we have the next slide, please? So pre-discovery process. If you want to make discover a drug, first you need to understand what exactly is the disease, and what is the underlying cause of the disease. What changes are happening in our body that it's causing such kind of untoward feeling in our body. What exactly changes are happening in the body? First, you have to understand the disease. Then our body is made up of cells, and cells are made up of something called genes, if you have studied biology. And all these genes, they express a particular type of protein. And there are various laboratory tests nowadays where you can just find out how your this various level of protein expression or gene expression is changing. And those changes, you can pinpoint at a particular reason that why are we getting this particular kind of disease in our body. So in previous days it was very difficult, but now with the medical science, like with the whole genome sequencing, RNA sequencing, single cell RNA sequencing, now we've got a lot of like advanced medical technologies, and now it's very easy to pinpoint what actual genes are involved in particular disease. And also there are, if that one particular gene or one particular protein that is expressed by the gene, how they are interacting with other cells in our body, and what kind of specific changes occur in our body. So in brief, the first step of starting a drug discovery process is you have to have a very detailed knowledge of the disease and what are the underlying causes of the disease. Can we have the next slide, please? So these are very technical terms, and some of you, if you are from a scientific background or a medical background, you will understand. So I won't go into detail explaining these terms, but I will just explain two terms: target and lead. What is target identification, or what is lead identification? Target is just what I explained in the previous slide. Is can be your gene or can be your protein. Suppose, for example, we take an example of cancer as a disease. Now, what is cancer? Cancer is there is an uncontrolled division of cells in our body. That division doesn't stop, and that's how people fall sick. So first you will understand why the cells are dividing in an uncontrollable manner. And once you know why the cells are uncontrollable manner, because you have found out a particular gene or a protein that is involved and that is causing the cells to divide in an uncontrollable manner. So that's your target. The gene or the protein, that's your target. So you identify that target. And what do you mean by validation and optimization? Validation and optimization is, suppose you have found out a gene that is involved in what causes the cancer to progress. Now the medical science has progressed so much with a lot of techniques. If you have heard about CRISPR, you can just simply delete that gene out of the system. Like, these experiments are actually done on mice first, or animals first. So you can just take out that gene, you can knock out that gene from animal, and see if the disease is progressing. Or you can also do it in vitro labs. I will explain you what in vitro labs are. So you can simply take out the gene from the body of the animal and see if the disease is progressing. So that's called target optimization and target validation. And what's lead? So lead identification is, once you have the gene which is optimized and validated, you need to find a candidate, a lead candidate, which can actually go and interact with the target. Which it can be, a target can be a receptor, and so you have to find a lead, which can be a drug, which can go and interact with your target and stop something untowards happening in the body. And that's called lead. How you define lead, and how do you identify lead? Now there are a lot of actually computer programs available, such as combinatorial chemistry, high throughput screening. Nowadays, you can just draw a structure in a computer software. A structure of the receptor is available, and you can just see if the lead, if the lead will go on and bind to the target, and whether it will produce any kind of effect or no. So, so, so first step of developing a drug is, you need to have a target, and after the target you need to find a lead which can go and interact with the target. Can we go to the next step, please? Next slide, please. So once you have the drug is synthesized in the chemistry, in a chemistry lab, and then you have the drug. Okay, so how we do that next? So once you have the drug, you don't actually go directly and give it to humans first. There have to be preclinical studies performed, and there are a lot of different animal species that are used in preclinical studies, which involves mice, rat, monkeys, zebrafish, rabbits, guinea pigs, hamster, and many more, even dogs. But nowadays, there is a lot of focus on reducing the use of animals, but still these are the common animals that are used in preclinical studies. So any drug that is newly identified and optimized, all those drugs are first tested in animals. This testing can be done in three ways: in vitro, in vivo, or ex vivo. What is in vitro? In vitro is doing the experiments in the lab. So you can just isolate the cells from the mouse or any other animal, or even humans. So you can take the cells out, and with proper conditions, you can grow those cells in your laboratory. And then you can add the drug to the cells and do some further downstream experiments to see if there are any changes. So that's in vitro. It's doing experiments outside the body, growing cancer cells, for example, in a lab, in a lab atmosphere. In vivo is doing experiments directly into the body, directly. Suppose if you want to study lung cancer in mice, you have got models, experimental models, where you can induce cancer in mice, specific to lung. If you just want another cancer, a cancer of kidney, leukemia, anything, you have experimental models developed. Even for arthritis, diabetes, you can induce any kind of disease in mice these days. That's how the science has progressed so much. So you just induce that disease in the mice first, and then you give your drug to the mice, and then you check if there is an effect. And ex vivo is, what you do is induce a disease in the mice, and then instead of giving the drug or something, you just isolate the tissues and do the experiment what you do in the in vitro settings in a lab, like grow those cells in an laboratory environment and do your experiments. People prefer to do ex vivo experiments because they are cheaper. than doing in vivo experiments. And as I said previously, there is a lot of focus on reducing the number of mice that are being used. So a lot of scientists now are actually encouraged to do a lot of ex vivo experiments and then go on in vivo experiments. And in preclinical studies, what you study is toxicology, is what is the dose that should be given, and if at that dose any side effects, what we call as adverse drug reaction or a serious adverse events are caused. So that we study in toxicology. Pharmacokinetics. So what is pharmacokinetics is, um, it's suppose you go to a doctor and a doctor tells you, like, you have to take one medicine in the morning, one medicine in the afternoon, and then one medicine at night. So why these three different doses, and why not three medicines at one time? So this comes to pharmacokinetics. What pharmacokinetics is that once you take a medicine, suppose a tablet orally, so that medicine contains your drug, and for that drug you have to maintain a steady plasma level or a steady blood level for your body to fight that infection or any other disease. So once you take the drug through your stomach, your intestine, the drug is absorbed into the body, then is distributed into different parts of the body. It will go to your heart, it will go to your liver, it will go to your pancreas, different organs, it will go to your lungs. And mostly the metabolism gets in the GI system, in the stomach and the liver. A lot of drug will get metabolized when we say is uh, broken down. Now what we when we eat food, the food is broken down in our body, right? So that's the same thing as the metabolism. The drug, when we ingest, it will get metabolized in mostly our GI system, in our liver, in our stomach, and other organs will also metabolize our drug to a certain extent. And then finally the drug is excreted. So how to decide how much dose is actually needed, whether we should take one tablet or whether we should take two tablets, is actually then decided in the pharmacokinetic studies. And pharmacology is actually looking at the effect. Okay, you have taken a tablet, but is it showing any effect? Or you have a headache and you take a tablet, but if is your headache going down? So there are what we call as various outputs which we define if your medicine that you have taken is actually causing any effect or not. Next slide, please. Then it comes as formulation. So most of the drugs, when they are in preclinical studies and when they are still tested in mice, the scientists are already aware many times what route um, what route and what form this drug should be given, because from toxicology and pharmacokinetic study, they have a very fair idea of how quickly the drug is metabolized or how quickly the drug is broken down in your system. So they have a very a good idea at the preclinical study level in what formulation that it should be developed for human use. So when I say formulation is, it can be a tablet, whether the drug should be given as a tablet, a capsule, as an injection, as an oral suspension, ointment, emulsion, lozenges. So these are all terms like you have for a non-medical person or a non-pharmaceutical person. These are all medicines. And like, why you get one medicine in tablet and why you get one medicine in capsule and one as a suspension or one as an ointment. So there are a lot of studies, and there is a lot of research that goes into place to decide what formulation that we should actually give this drug in. So lozenges is like, suppose if you have a lozenges, I will give an example of strepsils. Okay, so lozenges is for you need to have some local effect on your throat because you have an irritation in throat. So a doctor will give you a strepsils or a lozenge, which you don't take as a tablet or a capsule. You don't just swallow it. You keep it in your mouth. So whatever the medicine, the drug is present in the strepsils or in that lozenge, it will have a local effect on your throat. So that's why, what is the intended effect you need and what is the site. Suppose if you have a stomach pain, whether you have a pain in the bone, if you have a headache or anything, Your actual disease decides the type of formulation, and even that formulation you can take by various routes. It can be oral, it can be intramuscular when I'm talking about the injections that you get on the thigh or any other body part. For nasal, like you need a local application, like you have a nasal blockade, then you can inject directly the drug through your nasal inhalation. Ocular is through eyes. Intravenous injections when you have got a systemic infection, or if you take the drug by mouth, but it's not, it's very unstable in your stomach, so the drug will get quickly excreted from your body. So at that time, doctors prefer it to give through intravenous. And also intravenous, when the drug directly goes into the blood, it has a quick effect. So also to have a quick effect, the doctors will give you some drugs intravenously. And also topical, suppose like if you have a muscle pain or something, then you give it directly for a local action. You have an ointment, uh, which you just apply directly on the skin. And I've just shown a very small diagram over here of actual drug and the excipients. So when you take a tablet, so the first small drug you will see, first yellow, blue dot you see, is just the actual drug that is present, and the rest is all excipients. And what are excipients? They are mostly used for taste masking, stability, odor masking, appearance, to enhance the solubility, to have it a good flavor. There are various sweeteners added. Preservatives are added so the drug can be stored. It doesn't get degraded. So preservatives are added to um, so it can enhance its shelf life. So there are a lot of excipients that are added. Suppose if you have a paracetamol which says 500 milligram, so if you weigh the tablet will be a 1 gram tablet or even more than that. So actual drug is very small. The entire thing is made up of a lot of other things, and these are mostly to see that the core drug is stable in whatever form it is. It can be injection, it can be a capsule, whatever form it is, just to show that the drug is stable. And also, what are factors? Age. Your if it's a kid, a less than 5 year old, it's very difficult and challenging. They might choke on a tablet or a capsule. So mostly for children or young kids, doctors give oral suspension instead of a tablet or a capsule. So the actually the age defines. And as I said, site of action, if you have just a muscle pain, you won't take a tablet. You will take a Volini or something, which is a gel, and just apply on your muscle, and you will get a quicker relief rather than taking a tablet orally. Dose, frequency. I mean, I can keep on talking about these various factors that affect, but just keep in mind, like whatever medication you take, whether it is tablet or a capsule, whenever you take a medication next time, just have that in mind that a lot of research has gone into place, just to choose like why this particular drug is actually a tablet and not a capsule, or that why this is a capsule and not injection. So there is a lot of research that goes into it. Next slide, please. Clinical trials. So once the study they are finished in humans, in sorry animals, then you have to actually you see, okay, the drug is good, you take, there is a good action, like if there is a cancer, you can treat cancer. If there is a diabetes, you can lower the blood sugar levels. So that you kind of information that you have in the from the animal studies. So then you want to test those drugs into a clinical setting. So the clinical setting, I will just uh, directly go to the Nuremberg Code. Can we have next click, please? Yeah. So what is this Nuremberg Code? So in World War II, there was a lot of atrocities being done by the Nazi Party against the Jews, and the Jews people were used like animals to test their drugs. Like there were a lot of poison testing, injury testing, like gas chambers. They were tested directly on humans, and the humans or people, the Jewish people, they were used as animals in those times. So after there were a lot of court trials, which is actually called Nuremberg trials, and now from the Nuremberg trials, because of the atrocities done by the Nazi Party in World War II, they came up with this Nuremberg Code called in 1947. And there are 10 main points from this Nuremberg Code, out of which the most important is consent. That if any drug has to be tested on human, the person has to give a consent first, like, yes, I give you the consent to test this drug on me. The other thing is like, you can see the fourth point from the left is animal experiments first. You first test that drug in the animal, see it's safe, see it's effective, and then taste it on humans. Also, whoever is involved in the trial, in the testing of the drug, they have to be a qualified doctor. Even the support staff, they all have to be trained. They all need to have proper qualification to do these trials. Another one more thing is that if the subject or the patient is involved in the clinical trial, and he at any given moment of time he is not happy and wants to withdraw from the trial, he has to have the full right to withdraw from the trial. Like the subject should be free to stop. So there are a lot of other things that were involved in the Nuremberg Code, but these are the 10 things that were first designed, and that's what the Nuremberg Code is, and that's how the clinical trials from 1947 has been designed since 1947. Next slide, please. Okay, I'm seeing that there are a few questions, so we will take those questions at the end of the talk. I'll just finish my presentation first, and then we'll move on to the questions. So there are a lot of in clinical trials. There are a lot of aspects that have to be taken into consideration while designing a clinical trial, and there are four things which we call as a gold standard in a clinical trial. Every clinical trial should have these four things. One is blinding. What is blinding? Blinding is even the doctor doesn't know what medication that the patient is receiving, and even the patient doesn't know what medication is receiving. This is just to avoid any kind of bias in the results, like, so the data corruption is totally eliminated. So you can have a double blind, so the doctor knows, but the patient doesn't know. Or you can have a double blind, a triple blind. Like triple blind, when everyone involved in the trial, the support staff, doctor, and the patient, all three of them, they don't know which drug they are actually getting. So that's called blinding. All clinical trials should have a control arm and a treatment arm. So there will be some certain of populations which won't receive the treatment, but one set of population which will receive the treatment. So that's a control arm. Randomization is which arm of clinical trial will you go to it? Will it be a control, or will it be a treatment? So this is all nowadays generated by a computer software. You don't have to do much. This is once the patient is enrolled into a clinical trial, this is all computer generated. And so the entire clinical trial is guided by something what we call as a clinical study protocol. And what details does the study protocol involves? The study design, the inclusion and exclusion criteria. Not that every person who wants to take part in a clinical trial can be a part of the clinical trial. There has to be certain exclusion criteria, like, you shouldn't have any other disease. Sometimes there are weight criteria. You have to be healthy. You don't have to be having any other disease. Suppose, like, in a lot of clinical trials, if you are a heart patient, or if you have a diabetes, if you have a pneumonia or respiratory condition, then those kind of people are excluded from the trial, because they are at a greater risk. If there is any side effect, any adverse reaction of the drug, these people are at a greater risk. So there is a lot of inclusion criteria and exclusion criteria, what people should actually be in the trial. And once they are in the trial, what procedures will be done on them? Like, if they are going to do a blood examination, stool examination, are they going to be an X-ray, CT scan? What kind of examinations are being done on the patients? The schedule, what timing, at what time, 1 month after taking the drug, 1 week after taking the drug. So it's all very detailed. And before the person is enrolled into a trial, before a patient or a subject is enrolled into a trial, everything has to be explained to the patient. The study protocol has to be explained to the patient, all its pros and cons. They have to be made that fully aware that, like, you can't have very serious side effects with this. You can fall ill. Your disease might get worsened. They have to be made fully aware. The patient or the subject enrolling into a clinical trial has to be made fully aware. Next slide, please. And once the drug enters the clinical trial, there are various phases of clinical trial. Okay, phase one, phase two, phase three, phase four. So as you can see in phase one trial is a very is between a very small number of people. Okay, 20 to 80 people maximum. And here they just check the safety, if the drug is safe or not. They are not worried about the effect at that time. They just check the safety in the phase one trial. And below, just look at the success rate. 70% success rate. So out of 100 drugs tested, only 70% go to the next stage. 30% they fail in the phase one. That's it. The research is finished. All the preclinical studies, everything they have done, that goes into the bin. So 70% success rate. Okay. First phase one, the drug is safe. Then they go to phase two. Phase two are more number of people, 100 to 300 people. It can go up to a couple of years, like 2 years. And look at the success rate now, going sharply to 33%. Out of every 100 drugs that go to phase two, only 30% succeed. 70% of the drug, or 70 drugs, they go into the drain. So the success rate keeps on reducing as you go into the later phases of trial. It's just because you're testing them on more and more people. And then when you test the drugs on a wider population, or different kinds of side effects and everything is some kinds that the side effects are very serious. Sometimes there is not much effect seen. The effect is not that big. So the drugs keep on going out of the various phases of clinical trials. Then you go to phase three. Phase three is like the biggest trial. It goes on for several years. It says over 4 years. It can be go to 4, 5, 6, 7 years as well. But then that mostly takes sometimes time to recruit that many people, like 1,000 to 3,000 people. I think this is still less nowadays. The rules are so strict depending on the type of disease. Sometimes you even need 5,000 people, and that too not from one country. These clinical trials, they have to be global in nature, so they call it as a multi-center trial. Phase one, phase two, they can be done in just one particular country. Okay, but the phase three trial, it has to be global, multi-center trial. So if so there's a trial done in America, it has to be done in Asia, it has to be done in South America, it has to be done in Africa, because all these people, they will have a different genetic background. And it's very important to check that the drug still has the effect irrespective of the genetic background. So most of the studies these days are global multi-center trial, just to see the that the change in that genetic effect doesn't affect uh, the success of the medication. And the phase four is actually a post-marketing trial. So I'll just go explain it on the next slide. Can we have the next slide, please? So see over here, um, how the overall drug discovery process goes on. If you just go to an extreme left, drug discovery, this is the part when I'm talking about target identification and lead identification. Look at the number of compounds that you start with. 5,000 to 10,000 compounds you start with. And then from those 10,000, only 50 to 50 will go into a preclinical study. From that 250, only 5 will go into a clinical. This all then they all, like, they keep the rest of the drugs, they keep on failing at various stages of testing. And of those 5, which goes into a phase 1 clinical trial, ultimately only one passes the phase 3 trial. And look at the timeline, right from the pre-discovery stage. The entire drug discovery process, it's a very long period, 10 to 15 years. 15 years is what is generally take these days, 10 to 15 years to discover one medicine. And look at the cost. It's a very risky process. Three billion US dollars to discover one drug. And a lot of companies actually, they are protected then by patent intellectual property laws, so that no other companies copies their drug. But then it's a very long and a risky process. And what is phase 4? Phase 4 is once the drug passes the phase 3 trial, it receives all the approval, and then it goes into the market. But that market, like phase 3 trial, you just do 3,000, 5,000 people. Once in the market, millions of people start taking the drugs. And sometimes the doctors then, once the drug is taken by millions of people, the doctor realize, okay, the effect is not too much, or there are a lot of side effects. And even most of the drugs, after 1 year, in phase 4 of post-marketing surveillance, the doctors stop prescribing them, or what we see are they are rolled back. There are a lot of side effects, people are dying or something, then the drug is rolled back. So imagine spending this much amount of money, spending this much amount of time, and even in phase 4 the drugs are rolled back. So drug discovery and development, it's a very long road, it's a very expensive road, and it's a very risky road. Can we have the next slide, please? And this all drug discovery process is, like, I would just say, it's a collective effort. There are various branches of pharmacy, such as pharmaceutics, which looks into manufacturing, formulation, quality, packaging, chemistry, pharmacognosy, pharmacology, and pharmacy practices. These might be very alien terms to you. The reason I'm saying mentioning these terms is there's a lot of different disciplines in pharmacy which look and take care of the various aspects of drug discovery process. And when these all various branches of pharmacy, they work in sync, you have got one drug finally which comes into the market. That's it. Next slide, please. So yeah, thank you. That was my presentation. And I can see there are some questions. And if anyone wants to ask questions, we can just start asking questions now. So I can see Rupali is asking what is arthritis. So arthritis is a bone disease when your body's own immune system, it starts affecting your joints and bones. That's when it generally happens in old people. It's arthritis, what we call is an autoimmune disease. Your body's own immune system starts attacking your immune system, your joints and bones, and that's when you get arthritis. And the typical symptoms, like, what you say, you have, if it's a osteoarthritis, then you have difficulty walking. If you have arthritis in your hands, fingers, then it becomes very difficult to hold things, to move your fingers. So that's arthritis. Himangi is asking that if we are having allopathy treatment, then we should not go for homeopathy. This is something which I would refrain from answering. The only reason being, now this might be very controversial to a lot of people. I personally don't believe in homeopathy. I don't think that's a proper form of medication, and that's why I did not even mention it a lot of about me in my presentation, because for me personally, homeopathy is not a proper form of medicine or actual science of medicine, because it's just because what I talked about, the whole discovery process, the preclinical studies and the clinical studies, and even in Ayurveda these days, this whole drug discovery process doesn't take place in homeopathy. And there are a lot of drugs tested during using those these preclinical and clinical studies, the homeopathy drugs, and they haven't had effect which is claimed to be. So but if you believe that homeopathy is helping you, I wouldn't stop you from you taking homeopathy drugs. Yeah, you can. Ayush is asking if you can call allopathic medicine as drugs. Yeah, drugs is like, we have a very different imagination of drugs. But yeah, drugs is just a very small amount of substance that is present in your tablet or capsule, which we can just you call them as medicines. But like, in a general pharmaceutical or a very technical knowledge, we call it as a drug, or we also call it as an active ingredient. Tanmay is asking, can gene mutation be reason for different thinking from generation? That's a very interesting question. Um, I don't think so. Gene mutation shouldn't affect necessarily your thinking, not from generation. In your own body, if your thinking changes from from what your younger self to your older self, if your old thinker changes, a lot of there are diseases such as Alzheimer's or Parkinson's. It's when you become old, your brain also starts becoming old, right? So there are a lot of enzymes and hormones that are secreted in the brain, which can affect your judgment, which can affect your decision making capacity. And I think that with old age, as your brain also gets older, those hormones, those proteins that are made by brains, those also their level of expression changes, and it can affect your own thinking. But between generation, I don't think that would be true. Lead, how lead can help cure? Samyak is asking, and unfortunately, Samyak, I actually don't have answer to lead. I haven't come across lead as a cure. Lead can be actually can be poisonous to the body, but I have not really actually come across anything lead that can cure anything. Do drugs have a target organ? Yes, all drugs, they do have a target organ, they have a target disease. These days, the drugs are, what you can say, made in such a way, they are made intelligent that they would just go and find that target organ on its own, the way they are, what we call as a homing. So they can go into larger amount, or you can make it go to that particular organ and stay in that organ for a longer period of time, so that you have an effect. Is there any specific code in imparting color to the drug other than excipients? uh, yes, so there are a lot of colors, and obviously these colors are mostly natural in origin, and all those colors that are being tested as excipients, uh, they have been, stability tested, safety tested. So a lot of testing are done on these drugs before they are used in any tablets or capsules, and they are and it absolutely made sure that whenever you take a capsule that is a blue or a pink in color or a red in color, um, um, be assured that those are mostly of natural origin, or even if they are chemical in nature, they are absolutely safe for the human ingestion. And there is also a very specific set of chemicals or colors that is approved by various regulatory agencies such as US FDA or UK MHRA, which are the drug regulatory agencies in different countries. And they have a very list of the colors and excipients that you can use. So, yeah, I hope that answers your question. And what if the patient dies during drug testing? That's a very important question. And first of all, even in the drug, before enrolling in the testing, the patient is made aware, you might die. It is considered as the most serious adverse event. And if it happens, actually the company is not responsible for that, because uh, they are already making the patient aware, there can be very serious side effects. But the thing is that it's not that, okay, the drug is testing and the patient dies, okay. The patient might die of some other reason, not because of the drug. If the patient, met a car accident, was like, if the patient was alcoholic, he was doing some other sort of drugs, he did not have a healthy eating habit. So they do go and find a root cause of what actually the patient died. Did the patient die because of the drug, or did the patient die for some other reason? And still, what these days is happens is that, there is kind of an insurance money that is given to the patient, to the patient that is involved in the trial. Like, if the patient dies, the company does give some sort of money to the patient after the patient dies. But it's not the company's responsibility, because they are made aware right in the beginning of the trial that anything can happen to you. Sneha is asking, are there any drugs that can permanently cure the hormonal issues in women like thyroid and PCOS? Yes, the hormone therapy is like a very upcoming, I won't say upcoming branch of science, but there is a lot of research that is being done on hormone therapy. And yes, there are a lot of, especially like, in UK, when women go into menopause, there are a lot of hormonal changes that happen in women, and there is a lot of hormone therapy that is made available to older women in this country, where they can have this and the hormonal balance is still maintained in women body. So yes, there are a lot of therapies that are available. Permanent cure, I don't think so. I'm not sure. You might have to have frequent hormone therapy to have those level of hormone level in your body. So it's not like, you take the hormone therapy once and then you're cured. You might have to take a repeated dose of the hormone therapy to have those kind of steady levels of hormones in your body. I hope that answers your question, Sneha. uh, Akanksha is asking about vitamin tablets. So the only vitamin tablets, like, the UK government actually recommends is vitamin D, and that too because in this country, in UK, there is no sun. So and it's through sun that we get vitamin D, right? So the UK government actually recommends only one vitamin D. And what I personally believe is, if you're having a very good healthy diet, if you're having leafy vegetables, if you're eating fruits, if you're having eggs, breads, everything, if you're having pulses, or even meat, any non-veg food that you get, if you're having a good diet, if you have a good 3 times of meal, if you're drinking enough water and everything, your immunity will always be good. uh, the best thing is to have as much possible of green leafy vegetables. This has been, we have been learning this right from the schools. But now when I'm researching into things like cancer and everything, this is one of the most simple thing. Like, if you have proper balanced diet, like if you have fruits and leafy vegetables and pulses and eggs and everything, a balanced diet should give you all the vitamins, and you should have a very good immunity through a balanced diet. But if you think you are not eating well, I won't stop you from taking vitamin tablets. Actually, if there is some vitamin lacking in your body, they should help. But I would say they don't do any harm either. That's the thing. If you're taking the recommended dose, uh, they don't do any harm either. So, if you want to take vitamin supplements, you can take. And homeopathy, as I said, like, I don't believe a lot in homeopathy. I hope I don't offend anyone, but I'm not a great believer in homeopathy myself. This is just a very personal opinion, and I would need to have a very good conversation with a homeopathic doctor to try and convince me that homeopathy actually works. But again, as I said, this is a very personal opinion. Aditya is asking, is can drugs cause mutation in genes and chromosome? Yes, they do. They do. But a lot of this is um, taken care of before the drugs goes into, even in just in animal studies. Actually, they look at DNA damage and everything in animal studies itself, in preclinical studies. And even in preclinical studies, it's very evident that, like, this particular is drug is doing DNA damage or is causing any mutations in the chromosomes, and those drugs then actually don't go forward into any further research. Can we make simulators to test drugs? Preclinical testing. Yes, HVC support. Yes, we can. And actually, that's the thing. The overall scientific community and the overall research community is pushing for is, like, animals. No one like to test drugs on animals, and globally, the animal right activists, they have made it very difficult for pharmaceutical companies, even for academic universities, that they are forcing to reduce the number of animals that are used in preclinical studies. And so now a lot of focus or attention is given to is how we can simulate, and there are various ways uh, that you can simulate. And some countries like Germany, uh, they are thinking in like next 10 or 15 years they won't use any animals at all. Like, to that level they want to simulate the testing conditions, that they want to totally stop animal testing in next 15-20 years. So Tanmay is asking about gene therapy. Yes, Tanmay, gene therapy is actually the next big thing, and a lot of therapies are being developed. And the beauty of gene therapy is, so if you take a drug, the drug will go and act on your different parts of the body, even where you don't need the drug to act on. The drug and will go and act. But the beauty of gene therapy, it can be very site specific. It can be very specific to a particular organ. Not even an organ, like it can be very specific to a very part of compartment of an organ. So gene therapy will come into effect, but still I think it's when we have it's still a time to have a comprehensive gene therapy for each and everything to have. But yeah, that is the next big thing. Okay, Mega is asking, what if the discovered drug cured the root cause of the disease, but has other serious side effects? Okay, that's a very good question, Mega. Uh, so sorry. So whenever the drugs are being tested, okay, suppose cancer drugs, okay, but I'm constantly mentioning cancer because that's what I research on. Cancer drugs, okay, uh, the chemotherapy, what we call, uh, they have very bad side effects. Like if you have seen, you start losing your hair, you start losing your sense of smell, you start losing your sense of taste. You don't feel anything. It drains the body, it drains your energy, you're lethargic, you lose weight, you have headaches. Cancer has got a lot of side effects, right? The drugs, the chemotherapy, it has got a lot of side effects. But if the drug is curing the disease, suppose if I take a drug and it's curing my cancer, yes, I've got side effects. But then what we call it as a concomitant medication. So you give some other kind of medication to reduce those side effects. So suppose you take a drug for cancer, but then you get a headache. Then the doctor will give you a paracetamol to reduce your headache. So there are some other medication given to reduce those side effects. But in the end, the patient health is the most important in those clinical studies. And if there are too many side effects that are being observed, then obviously the drug will fail in the phase 1 trial or the phase 2 trial, and it won't go into the phase 3 trial. So and there is an always of benefit risk analysis. How much are the benefit and what are the risks? Do the benefits outdo the risk? Yes, then the drug should go. If there are the risk outdo the benefits, no, the drug will not go into the later phases of clinical trials. Hope that answers your question. uh, Dr. Nagabhushan is asking, can you throw some light on Ayurvedic preparation which is being practiced since ages? My opinion of Ayurvedic medications is, I think they are very good preventive. They have got very good preventive properties. Treatment. One thing that the West that have not accepted Ayurvedic medication as much as they should have been is because our ancient sages and even now the whatever the research has what we have in Ayurvedic medication, the West doesn't accept Ayurvedic medication because all those the phases of testing that I talked about, the preclinical stage, the clinical stage, and all this money, our Ayurvedic medication, they are not scientifically validated, right? And it's an expensive process. Like for any company or anyone to do the scientific studies, it's a very expensive process. And because of those lack of scientific data, there has not been a lot of acceptance of Ayurvedic as a treatment. Okay, as a good way of living, yes, I think Ayurveda is very good. As if you want to change your diet, like there are a lot of things like Ayurveda mentions, like okay, you should have hot water with a turmeric in it, and like suppose if you've got cough and cold, you should have a haldi and doodh, warm haldi and doodh. These are very effective treatment, and obviously they have some effect. So to prevent yourself from disease and just to treat like normal disease, like cough, cold, some pain, headache, if there are some Ayurvedic options available, you should go because they are natural. That's the thing. With allopathic medicines, they are all synthetic chemicals, like they are chemicals made in the lab. So that's the advantage that Ayurveda has over allopathic. But like, if someone comes and say that, okay, Ayurveda will treat your cancer, if Ayurveda will treat your diabetes, and Ayurveda will treat your osteoarthritis, I don't believe it, unless and until I have an extensive unless and I have an extensive studies that are proving that, like, this Ayurvedic medication is treating my cancer, I won't believe it. So all the medication have to be scientifically validated. Is apoptosis responsible for Alzheimer's? Yes, it is responsible for Alzheimer's. Alzheimer's is a neurodegenerative disease. Your cells, they die over the period, and yes, that's what causes Alzheimer's. Aditya is asking. That's true. Akanksha is asking, is there are many assumptions about vaccination on corona? What is your opinion? Corona vaccine, like, there are a lot of companies, they are involved in vaccination. Like, I'm working at University of Oxford, and we have been developing a vaccine, not me in particular or my team, but I know certain people, they are involved in vaccine development. And any vaccine, it will be an another year, because I told you, right, the various phases of clinical testing, phase 1, phase 2, phase 3. So there are three, and now that process is like 6, 7, 8 years. Now, because this is a pandemic, people are saying, like, the government will fast track everything, and we might have a vaccine in 1.5 year. But that testing, like 1 year, will have no vaccine at all, even after 1 year, because what if it fails? Like, it's other thing, testing the success of a vaccine in preclinical studies. Like, the corona vaccine, what mostly they are doing is they are testing in monkeys. So how they test is they give a small virus to the monkey. So the monkey gets coronavirus, okay, and then no, sorry, the other way around, they give the vaccine first, um, to the monkey, and then they give a small dose of virus to the monkey, and see if the monkey is protected or not. So now the initial study that University of Oxford did was on monkeys, and I think, if I'm not wrong, they did this study on 28 monkeys. They gave the vaccine, and then they gave the virus to the monkeys, and all the monkeys survived. None of the monkeys fall ill. Um, so and now that's how it is into humans now, but it's still a very long process. If it works in monkeys, it's not necessary that it will work in humans, and it's still a very long time, and even after 1 year we won't have a vaccine. It's as possible as that. What if receptor on cell membrane CD4 is absent? Naturally, I don't think it should happen. It should your body just won't. Aditya is asking this question, and I don't think your body will just naturally lose all of its CD4 receptor. It might be reduced, but not absent at all. I don't think that will happen. Are there any other questions? I'm happy to take more questions.
Yeah, okay. So even if you don't want to type questions out, if you are on a PC or on a Mac, maybe using Chrome, you can just raise your hand and I'll just take you on the stage. You can ask it live. Okay. Also, after the session, for like about 15 minutes, the speaker will be available on table number 1. In the social lounge, you can just come in and interact. Okay. So if there are doubts, like any doubts, you can raise your hand. Also, I'd like to mention for college faculties, we are taking in affiliations. So your college can be affiliated to our club, and we can arrange some more webinars for your college. So I'm just leaving a link in the chat section. You can just maybe fill out that form if you are a college authority, or you can just forward this form to your seniors or anything like that. So you can affiliate with our club. Okay, so
Okay, I
maybe
think,
we should
yeah,
end the session. Okay.
yeah.
Yeah. So as I said, you can go into the social lounge on table number 1. The speaker will be there for about 10-15 minutes. So you can just interact with us there. I'll be on table number 2. If there are any doubts about affiliations, you can talk to me there. Okay. So thank you very much, Dr. Mahale. I'll just end this session and we'll meet in the social lounge. Okay.
Okay, thank you.